Bismillahirrahmanirrahim. Dear students, Assalamu alaikum and welcome back. In the previous video, we discussed formation of peptide bond. Uh, just a quick review. Let's draw a bond, peptide bond again. So we have two amino acids. Suppose this is amino acid number one. So we have alpha carbon NH2 on one side. Uh, this is C double bond O, and let's draw it in a different color and make it a little thicker. And now let's draw the other amino acid. So we have N here, H, C, H, C, and double bond O, OH, and R. And this is the R of amino acid number one. Now this particular area in the center, this one, this is the peptide bond. In chemistry, uh, this is usually known as amide linkage. So amino acids in proteins are united by amide linkages, which we commonly call the peptide bond, and they join in a linear fashion. Now the next amino acid is going to bond to this particular amino acid from this side. So let's draw, for example, so we have N, H, H, alpha carbon, R, H, C, double bond O, and OH. Now what's going to happen next is that this particular area and these two, they are going to form a bond. The hydroxyl group and H group from these two amino acids will be removed, and nitrogen and carbon from the two amino acids will bond, and we are going to get another peptide bond or amide linkage. So this is how amino acids bond, and as you can see, this bonding is in a linear fashion. It will continue as long as uh, the peptide chain allows, or the number of amino acids that may be present in the, if it is 100 amino acids polypeptide chain, then it will end at the 100th amino acid and so forth. Now let's take an example of some real amino acids. So we are going to make peptide bond between three different amino acids, and these are in fact real amino acids. And these are histidine, cysteine, and valine. So let's go through the process uh, really quickly. So let's draw the first amino acid, histidine, which you remember we have drawn in one of our videos of today, the basic amino acids. So we have C, which is the alpha carbon. This is the H, the N terminal, Now here we have the R for histidine. If you remember, it is, now we are not going to draw all the bonds, CH2, and then we have a 5 cornered ring. Let's draw a different color. So we have C here, C and nitrogen, and then in the lower corners we have N and C. So this is basically a 5 cornered ring with double bond at these two locations. Add hydrogen atoms to all of them. Now since here we have 4 bonds, so it gets a positive charge. And also we have an H here, so it also carries a positive charge. So this is amino acid histidine. Let's draw the peptide bond. Sorry. Uh, Next will be its carboxylic group, so O, and here comes the peptide bond. So now it's the next amino acid, and its amino group, and then we have its alpha carbon. It is its hydrogen atom. In the side chain, slightly different. We haven't drawn these amino acids. This is cysteine or cysteine, the sulfur carrying amino acid. And here comes its C. Let's draw it the other way. C double bond O, and then comes the next peptide bond. So first amino acid is histidine, the second amino acid is cysteine, and the third one is valine. So again we have this nitrogen here, then H, the alpha carbon, and hydrogen on one side, and on the lower side we have the R. So C, H, and if you remember in valine we have 2 methyl groups attached to this carbon. And then on its right is C double bond O. Actually, the hydrogen from this hydroxyl group, if you can see, we have it here, 3 hydrogens on the amino group of the first amino acid. So as you can see, let's zoom out a little so that we can see the whole structure. Now we have 3 amino acids bonded. Now we didn't draw the full amino acids or the formation of the bonds. We have drawn 3 amino acids with the bonds already formed. So let's see what we have here. So this particular area, as you can see, this is one of the peptide bond area, and this is the second peptide bond area. As we have discussed in the previous video, that these areas, uh, they are rigid and they do not allow any movement. However, uh, the bonds in between these two areas, and these are the bonds here. Now these bonds allow rotation, and since obviously when we make a new bond uh, between two amino acids, then we have more bonds which can rotate, and rotation of these bonds allows the polypeptide chains of amino acids very flexible, so they can bend in any direction, uh, allowing the polypeptide chains to fold in various fashion. Obviously it depends upon the R chains of the uh, amino acids. So in this video we have seen bonding three actual amino acids, and we have also seen the peptide bond area which is rigid. It doesn't allow any movement. However, the area between the two peptide bonds, similarly when we make new bonds, so the area between those two bonds, and that will be in this particular place. Next one will be formed on the other side. So this area, as you can see, the side chain and where hydrogen atom is located, this particular area. When a new bond is formed on this side, so this area will allow rotation, free rotation, and this makes the polypeptide chain very flexible. So this was the formation of peptide bonds between three real amino acids, the location of the peptide bond which is rigid, which has been shaded in pink, and the bonds which allow rotation of the polypeptide chain so that it becomes flexible and allows folding, as we shall discuss in our next videos, the secondary, tertiary structures of the proteins. Thanks for watching, and see you in the next video.